Hello, my name is Sabrina Templo. This video is for Bio 225. Today is May 30th, 2020, and this is my results video for my lab of bacterial morphology and staining techniques. So I'm going to talk about the process that I did. For exercise 1, I used my gum and teeth swabs to make direct and indirect slides. So the first one I did was a direct slide, and I did a simple gram stain, and I used the crystal violet gram stain, and it produced a purple color, and you could see it through the microscope. And then the next one was indirect, and you use the Congo red. So unlike the first direct one, and instead of heat fixing the bacteria to the slide and then adding the dye, so what you do is you add the Congo red to the slide, and then you take your swab sample and then agitate it in there and pull it with another slide in a 30-degree angle and wait for it to dry. It was kind of cool because you're looking at the same thing, but the technique is different, and it's cool to see that you also got different results and what each bacteria looks like. So the next exercise was the gram staining. So you used all 4 stains. So the first one we used with the S. cerevisiae, and just going back that I did do my whole lab. In the lab instructions, I was so confused because it looked like there was so much color on the slides, and I already did all my slides during the experiment, and I was kind of confused because I was comparing my slides to the instructions, and I was thinking that it should have more color on them, but I did put it under my microscope. But I did put it under the microscope. I did see some bacteria, but then I did it all over again, and I did get the same result. But it was kind of cool to compare each of the slides. And so going back, I have the S. cerevisiae, the E. coli, and the S. epidermidis slide. Because the procedure was like you make sure everything is aseptic, which was very important, and putting the broth onto each slide and wait for it to dry completely, and you do the heat fixing, which helps the bacteria stay on the surface. And then you do the gram staining, which first one you do is the crystal violet stain. And basically what this does is no matter if it's gram positive or gram negative, it's going to stain all the bacteria like a nice purple color. So basically you leave the crystal violet on for about 60 seconds, and then you wash it off, and you put it back in the staining tray. And once that's done, you do the second one, which is the VP iodine. And it's like a orangey red burgundy color, which is also used in hospitals for aseptic purposes. But basically what the iodine does is it helps the crystal violet stick to what we already have. So it helps the crystal violet. So no matter if you have a gram positive or negative, it's gonna further solidify the color on the slide. And then after you leave this on for about 60 seconds, and then you wash it off. And then next is the decolorizer. The decolorizer strips off the color. The gram positive stay purple, and then the gram negative washes away. So that's what happens with the decolorizer. and then you leave this on for about 30 seconds. And then next is the safranin, which turns the gram negative, turns it pink. So the gram positive cells stay basically the same, and then the gram negative stays turns pink. So when this happens, what I learned is as opposed to a simple dye, just one dye, then you could do the whole set of the gram staining with all of these four. Allows us to see the different types of structures, no matter what cells, what dye you use. You can see under the microscope, which is, I found it was cool, which was different structures under the microscope. Again, I initially thought I was doing it wrong. I did do it a couple of times, like I previously mentioned, but I feel like since I did see bacteria under the microscope, that I did do it correctly in the end. And I found it difficult actually taking photos on the microscope, since my hands aren't that stable, so I was shaking a lot, so my photos might kind of look kind of blurry or streaked, and that's one of the difficulties that I found. Other than that, the gram staining process was a little bit tedious, but it was fun to learn and actually do the experiment. And that concludes my video for this week. Thank you.